Thank you, and thank you for the opportunity to present here today. So Actinogen Medical is an ASX-listed biotech company developing new therapies for cognitive impairment with Alzheimer's disease as the key indication, but we have a number of other indications under development as well. The our lead drug, Xanamem, has a novel mechanism of action, as do all of our drugs, and they're related to the inhibition of the 11-beta-HSD1 enzyme, an intracellular enzyme that activates cortisol at the cellular level. And what's been shown over numerous epidemiological studies now over some years is that chronically raised cortisol is associated with cognitive impairment and, more specifically, the progression and the development and the progression of Alzheimer's disease. The hypothesis behind our drug, the hypothesis behind the development of our drug, is that by inhibiting that cortisol production and targeting it at a cellular level through inhibiting the 11-beta-HSD1 enzyme, by inhibiting that enzyme, we're able to bring down the cortisol and moderate cognitive impairment and, importantly, Alzheimer's disease. The epidemiological data, as I said, has been produced through a raft of different studies. One of the more significant ones, produced by the ABLE group, demonstrated that in an elderly population, cognitively normal elderly population with a raised cortisol, there was a much higher risk of developing Alzheimer's disease. And what they showed as well is that 65% of people over the age of 50% of those over the age of 65 had a raised cortisol. So what we have is a convergence of aging, a raised cortisol, and the development of Alzheimer's disease, leading to the hypothesis behind our drug and evidence that has been supported by both animal models and human models, and now more recently data from a study demonstrating that this hypothesis, this inhibition of the enzyme and suppression of cortisol production produces an improvement in cognition. So we have a novel mechanism of action quite clearly for the treatment of Alzheimer's disease. We have proof of concept in animals. We have that data from many years ago. Proof of concept in humans from a pilot study many years ago and demonstrated just recently, and data that I'll show you. We've got a good, strong safety profile from our studies and excellent dosing data. So all in all, we're in great shape to initiate phase 2 studies. Just looking at the achievements over 2019, very significant achievements over 2019. The first one, the Xanamem trial, a safety study, standard safety study, 40 patients, 42 patients, but we included within that an efficacy component, and very strikingly and very pleasingly demonstrated a statistically significant efficacy outcome in just that small study. 30 patients on active drug, 12 on placebo, and we produced a statistically significant result. That was at 20 milligrams a day, and it's important to note that this is a dose twice the dose from that utilized in our phase 2 trial, Xanadu, that you'll see further in this slide. Also this year, we announced, or we are producing and announcing, data from our target occupancy work and homogeneous binding data studies that are demonstrating optimum dosing, clearly demonstrating brain penetration of our drug and occupancy of the 11-beta-HSD1 enzyme in the brain, and importantly, providing us with optimizing the dose that we will take forward into future studies. The Xanadu trial, a phase 2 trial, a large international phase 2 trial that read out in May this year, 186 patients. This one dosed at 10 milligrams a day. So there is a difference between the Xanahes and the Xanadu trial. The Xanadu trial demonstrated very good pharmacodynamic data, good safety data, but missed the primary endpoint. When we delved down into the data, we found a lot of signals, but not enough to actually indicate where we may have missed the efficacy endpoints. What's clear now from the data that is being generated is that it appears to have been a dosing issue. We may have underdosed in that trial, and potentially we require a bi-daily dose. We've also run a large animal cohort of toxicology studies, the longer-term toxicology studies required by all regulators, and those are progressing really well and reading out as expected. So a very productive year this year, and importantly, a year that has produced data that has given us confidence that our drug works, we're going in the right direction, and we're well positioned to now move forward into future phase 2 trials. The efficacy data that I was talking about from the safety study is demonstrated partly in these graphs over here, showing clear separation of the efficacy curves. This was a cog-state battery of efficacy that was run in the small safety study. Clear separation of the curves, separation occurring at week 2, certainly by week 4, there is a very clear separation, and that separation continuing out to the end of treatment at the end of week 12. It's persistent throughout and rapid. Data that replicates the early animal data that we had and the early human data. So here we have confirmation that that early data is replicated in an elderly human population. The actual data points for that trial are demonstrated here. You can see on your left-hand side the p-values showing clear statistically significant effect, and with the working memory, a highly statistically significant effect. But what I think is particularly telling is the treatment effect that is seen. Treatment effects, figures of 0.6, 0.7, 0.8, and one needs to balance that against figures that were achieved for Alzheimer's of 0.2 or vortioxetine 0.3. Now, in no way is that a comparison, but it shows that this is a very strong treatment effect that we are achieving with our drug in a very small patient population. At the same time, we're running a target occupancy study, a series of target occupancy studies, radio labeling a tracer. We had to develop a separate tracer, radio label the tracer with carbon 11, and the top line of this slide demonstrates the occupancy of the enzyme by that tracer. The bottom slide demonstrates after 7 days of treatment with Xanamem, in this case 20 milligrams, demonstrating the very clear occupancy of those binding sites by Xanamem and the inability of the tracer to bind in this cohort on the bottom. Clearly demonstrating a very good target occupancy of our drug, clearly demonstrating brain penetration of our drug as well, of course. We've done different cohorts of patients at 5 milligrams, 10, 20, and 30, and seen between a 50 and 85% occupancy at these different doses depending on the patients involved. We have patients, both healthy volunteers and Alzheimer's patients in the trial, so a very rich data source confirming that our drug gets into the brain, binds to the enzyme as expected, and achieves a very good target occupancy as designed. We're equally doing delayed PK to demonstrate the potential for a divided dose going forward. So in the end, through this and through a in vitro homogeneous binding study that we're doing with fresh human brains, a study being done in the UK, we are demonstrating the optimizing the dosing for our drug going forward. So a very productive set of data this year, which brings us to the point that we now have cognitive efficacy at 20 milligrams. Very robust data demonstrating cognitive efficacy at 20 milligrams in healthy elderly volunteers. We have target occupancy demonstrated between 5 and 30 milligrams. We have cortisol inhibition, data that I haven't been able to show you, but we have data to show that through both the cortisol-related hormones, so ACTH, DHEA, etc., we get good cortisol inhibition, and in fact we get a statistically significant peripheral cortisol inhibition at 20 milligrams a day. And we've got a good, strong safety set of data being generated. So we're in great shape to move forward, and this is the way we plan to move forward with our drug in Alzheimer's disease. On your right, you'll see the cohort, the healthy elderly cohort, where we demonstrate dosed at 20 milligrams a day, where we demonstrated that good efficacy signal. In the mild Alzheimer's disease population dosed at 10 milligrams a day, we failed to get the statistically significant signal that we expected. But sitting between them is mild cognitive impairment, probably the key target for most Alzheimer's companies, hoping that we are able to not only diagnose but treat Alzheimer's early enough to make that true impact on the patient before the neurodegeneration has taken over. A very intriguing trial that we're planning at the moment with our key opinion leaders and with regulators, and this is a study that we will initiate at 20 milligrams a day. But alongside Alzheimer's disease, the mechanism of action of our drug points to the opportunities to treat cognitive impairment in schizophrenia, cognitive impairment in bipolar, in diabetes, and a number of other conditions. We've had to undertake, or we have undertaken a deep dive, strategic deep dive on all of these opportunities, and have really come out with 3 main opportunities to focus on, and those are studies that we will initiate this year. The Alzheimer's trial, the mild cognitive impairment study, as I've indicated. Cognitive impairment in schizophrenia will be the second study we initiate, and a third one being cognitive impairment in type 2 diabetics. All of those populations demonstrating quite clear cognitive impairment associated with raised cortisol. But on top of that, there are other intriguing opportunities that we are looking at, and we will initiate studies in those areas as well. So strategically, we're in a great position to move forward. We're undertaking a comprehensive review of all of this data we've generated over 2019, refining the clinical development plan to include mild cognitive impairment in the future Alzheimer's trial, cognitive impairment in schizophrenia, in diabetes, and other studies as indicated. Grant applications are already in to support a number of these studies. Key opinion leaders and regulators obviously are being brought into the planning and the discussion around the trial design, and importantly, as we are doing here today, we're speaking with Big Pharma, who are very interested, very intrigued with what we're doing, speaking with Big Pharma about partnerships and collaborations to assist us in the development of both these indications and other indications, and recognizing that going forward into phase 3 and beyond, we would benefit from utilizing the expertise of Big Pharma. Importantly, as indicated on the slide here, at ADPD in Vienna in April, we will be presenting our data. We've been accepted for an oral presentation of our data, and that would include the Xanamem data, the higher dose safety data, demonstrating the efficacy that you saw, the target occupancy data and homogeneous binding data, and the Xanadu phase 2 Alzheimer's trial data. So a very comprehensive data set will be presented at ADPD. At the same time, we are currently submitting an abstract for AAIC, and we have in Amsterdam in July, and have every expectation of being accepted for an oral presentation for that as well. So the final slide is really a wrap-up of what I've been talking about, that really Xanamem and the development program we're undertaking presents an intriguing investment opportunity, both for investors and for collaborators, in taking the development of Xanamem forward in both Alzheimer's disease and these other indications. We have a differentiated compound, quite clearly a differentiated compound. We've demonstrated that it's efficacious. We've demonstrated that it's brain penetrant, and that it's a very selective, highly selective inhibitor of 11-beta-HSD1. Importantly, it's an oral compound. We've shown very robust cognitive signals, as you've seen, and we're moving forward into mild cognitive impairment in Alzheimer's disease and cognitive impairment in schizophrenia and diabetes as the next round of studies that we plan to initiate. All of the study parameters related to dosing, dosing frequency, dosing intervals, all of that is now, will be confirmed by all of the data that we're generating. So by the time we're ready to start these studies, all of that will be confirmed. We have a number of meetings and collaborations and discussions ongoing with Big Pharma, and we're very hopeful that Big Pharma will come on board with collaborating with us in the development of our drug. Having said that, we are well capitalized. We're a public company. We're well capitalized to take this program forward by ourselves and alongside the non-dilutive grants that we've applied for. But most importantly, the areas that we're working in, Alzheimer's, cognitive impairment in schizophrenia, cognitive impairment in diabetes, and some of the other as yet undisclosed indications that we're looking at, all represent huge unmet medical needs, and that are unsustainable with current healthcare budgets, presenting a magnificent opportunity moving forward to capitalize on this research that we're doing. Thank you for listening. Yes, as indicated, our biggest shareholder, in fact, is Biotechnology Value Fund, BVF, from San Francisco. They have quite a long track record of investing in early stage startup Australian biotechs, and a very successful record in doing so. And I must say it was very pleasing to attract them onto the register when we undertook a capital raise about 2 years ago. Understandably, they did a very detailed due diligence and examination of the data, and invested in us, and through all the highs and lows over the last 12 months have remained absolutely committed to us, and are still very enthusiastic supporters of what we are doing. And I'm pleased to say one of the meetings we will be having is with them to update them on the data that I've presented to you today. Alongside of them, Edinburgh University is our second biggest investor, and I have to say the technology came out of Edinburgh University, so that was an equity deal, and equally they have been very enthusiastic supporters of what we do, and continue to be represented on our scientific advisory board alongside our clinical advisory board, made up of global Alzheimer's specialists.
Thank you.
Thank you.